Sample question paper, class 12, code 044, biology 2024-25. Maximum marks 70, time 3 hours. General instructions. Friends, you can pause the video and see all the general instructions. Section A. Question number 1 to 12 are multiple choice questions. Only one of the choices is correct. Select and write the correct choice as well as the answer to these questions. Question 1. Signals for parturition in human female originate from answer B. Both placenta as well as fully developed fetus. Question 2. To produce 1600 seeds, the number of meiotic divisions required will be answer B. 2000. Formation of one seed requires fertilization between one pollen grain and one egg. To produce 1600 seeds, 1600 pollen grains and 1600 eggs will be required. Each microspore mother cell results in the formation of 4 pollen grains after one cycle of meiotic division. So, 400 meiotic divisions will result in the production of 1600 pollen grains. One megaspore mother cell after one cycle of meiotic division results in the formation of one egg. So, 1600 meiotic divisions will take place to form 1600 eggs. Thus, total number of meiotic divisions required for the formation of 1600 seeds will be 400 plus 1600 is equal to 2000. Question 3. A sample of normal double-stranded DNA was found to have thymine content of 27%. What will be the expected proportion of guanine in this strand? Answer A. 23%. According to Chargaff's rules, in DNA A is equals to T and G is equals to C. Thus, A plus T plus G plus C is equals to 100. Given T is equals to 27%, so A is equals to T is equals to 27%. Thus, A plus T is equals to 27 plus 27 is equals to 54%. Thus, G plus C is equals to 100 minus 54 is equal to 46%. Since G is equals to C, so G is equals to 46 divided by 2 is equals to 23%. Question 4. Observe the schematic diagram that depicts a small section of nucleic acid. The bases in two strands are paired through hydrogen bonds that are shown by the dark lines. Identify the correct sequence of nucleotide in the 5-3 direction. Answer B. CGTA. For visual impaired students, E. coli has 4.6 into 10 to the power 6 base pairs and completes the process of replication in 18 minutes. Then the average rate of polymerization is approximately answer 4000 BP per second. It completes replication process in 18 minutes, that is 18 multiplied by 60 seconds. Rate of polymerization 4.6 multiplied by 10 to the power 6 BP divided by 18 multiplied by 60 second is equals to 4,60,000 divided by 108 is equals to 4259.1 BP per second or approximately 4000 BP per second. Thus, the correct option is B. Question 5. Suresh and Rajesh have defected hemoglobin due to genetic disorders. In Suresh, the problem is qualitative as he is having incorrectly functioning globin molecules, while in Rajesh, the problem is quantitative as he is having very few globin molecules. Identify the disorder they are suffering from. Answer C. Suresh, sickle cell anemia, autosomal linked recessive trait. Rajesh, thalassemia, autosomal recessive blood disorder. Question 6. In E. coli, the lac operand gets switched on when lactose is answer A. present in the medium and it binds to the repressor. Question 7. Which of the following features shows the mechanism of sex determination in honey bee? Answer A. First and second. First is an offspring formed from the union of a sperm and egg develops as a female. Second, males have half the number of chromosomes than that of female. Question 8. The following diagram shows a fragment of DNA which is going to be transcribed. The upper strand with the polarity 3 to 5 is the template strand. After transcription, the mRNA can be represented by answer C. 5 UAA-CGG3. Question 9. Idli dosa dough rises due to production of which of the following gas? Answer B. CO2, that is carbon dioxide. Question 10. Adaptive radiation leads to which of the following? Answer D. Rapid divergence of traits among populations inhabiting a given geographical area. Question 11. ECO R1 cuts the DNA between bases G and A only when the sequence of GAATTC is present. The number of nucleotides present in the resultant sticky ends that will be formed in each of the two strands of DNA after this enzyme cuts the DNA will be. Answer A. Vector DNA 1 and 5, foreign DNA 5 and 1. Question 12. During the secondary treatment of sewage, which of the following change in the effluent occur due to flocks? Answer A. Reduction in BOD. Question number 13 to 16 consist of two statements, assertion A and reason R. Answer these questions selecting the appropriate option given below. Question number 13. Assertion A. Cells of tapetum have more than one nucleus. Reason R. They undergo meiosis without cytokinesis. Answer C. A is true but R is false. Question 14. Assertion A. Deoxyribonucleoside triphosphates serve dual purposes. Reason R. They act as proofreaders and provide energy. Answer C. A is true but R is false. Question 15. Assertion A. A floating cover placed over the slurry in a biogas plant keeps on rising. Reason R. This cover keeps on rising due to the gas produced in the tank by the microbial activity. Answer A. Both A and R are true and R is the correct explanation of A. Question number 16. Assertion A. DNA fragments can be isolated by gel electrophoresis on the basis of their size. Reason R. The larger the fragment size, the faster it moves. Answer C. A is true but R is false. Section B. Question number 17. Attempt either option A or B. A. First, a blood test reported negative for HCG. What does negative HCG imply? Name the tissue which produces HCG. Answer. Negative HCG implies no pregnancy and the tissue which produces HCG is placenta. Second, if a blood test reported positive for HCG in a person, then which other hormones would also be secreted by the tissue secreting HCG? Answer. Human placental lactogen HPL, estrogen, progestrogens, relaxin. You can write any two. Or question 17b. First, the human male ejaculates about 200 to 300 million sperm during a coitus. However, the ovum is fertilized by only one sperm. How does the ovum block the entry of additional sperms? Answer. A sperm induces changes in the zona pellucida membrane on contact, blocking entry of other sperms. Second, all copulations will not lead to fertilization. Why? Answer. Ovum and sperms should be transported simultaneously to the ampullary region for fertilization. Question 18. Attempt either option A or B. A. The schematic representation given below shows the DNA strand and two types of mutations in the DNA strand. Original template, mutation 1, mutation 2. First, identify the type of mutation exhibited in first and second. Answer. First is point mutation, second is frame shift. Second, which of the above mutation is more harmful? Give reason. Answer. Second, as more codons are affected. It is extremely likely to lead to large scale changes to polypeptide length and chemical composition, resulting in a non-functional protein that often disrupts the biochemical processes of a cell. Incorrect amino acids are inserted. Often premature termination occurs when a nonsense codon is read. Frame shifts have very severe phenotypic effects. Or B. Given below is a schematic representation of a mRNA strand. First, in the above sequence, identify the transitional unit in mRNA. Answer. Transitional unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon UAA and codes for a polypeptide AUG, AUC, UCG, UAA. Second, where are UTRs found and what is their significance? Answer. Untranslated regions UTR. The UTRs are present in both 5 end before start codon and at 3 end after stop codon. They are required for an efficient translation process. Question 19. Given below is the relationship between the HIV levels in the blood and helper T cell count in a person detected with AIDS. Study the relationship and answer the questions that follow. Concentration in blood, time, in months and in years. Virus cultured from blood, T cell count. A. What kind of relationship is observed in the virus levels and the immune response after some days of the initial infection? Answer. As the adaptive immune response gears up, there is a reciprocal relationship between virus levels in the blood and helper T lymphocytes levels. As the level of helper T levels rises, the virus levels decline. B. Does it completely clear the virus from the body permanently? Give reason for your answer. Answer. Several years later, if untreated, HIV patient will lose the adaptive immune response, including the ability to make antibodies, as gradually the HIV enters the helper T lymphocytes, leading to a progressive decrease in the number of helper T lymphocytes. For visually impaired students, write the sequence of events that occur when a retrovirus enters a human being, causing reduction in helper T cells. Answer. After getting into the body of the person, the virus enters into macrophages where the RNA genome of the virus replicates to form viral DNA with the help of the enzyme reverse transcriptase. The viral DNA gets incorporated into the host cell's DNA and directs the infected cells to produce virus particles. Macrophages continue to produce virus particles. In this way, they act like HIV factory. Simultaneously, HIV enters into helper T lymphocytes (Th), replicates and produces progeny viruses. The progeny virus released in blood attack other T lymphocytes, leading to a progressive decrease in the number of helper T lymphocytes in the body of the infected person. Due to decrease in the number of helper T lymphocytes, the person becomes immunodeficient. Question 20. A culture plate of lactobacillus shows blue colored colonies and colorless colonies. Explain the principle involved in the formation of such variance in the color of colonies. Answer. The variation in color of colonies is due to the principle of insertional inactivation. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the enzyme, which is referred to as insertional inactivation. The presence of a chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the beta galactosidase and the colonies do not produce any color. These are identified as recombinant colonies. Question 21. Attempt either option A or B. A. First, it was estimated that if an evergreen forest has a GPP of 400 and 150 worth of carbon dioxide flows out of that forest, what is the NPP in that forest? Answer. NPP is equals to GPP minus R. Given GPP is equals to 400, R is equals to 150. So NPP is equals to 400 minus 150 is equals to 250. Second, explain why pyramids of energy must always be upright. Answer. Pyramid of energy is always upright. As energy flows from one tropic level to the next tropic level, some amount of energy is lost in each tropic level in the form of heat. Therefore, the pyramid of energy is always upright and can never be inverted. Or B. First, assume that GPP forest A is equals to GPP forest B is equals to GPP forest C. If forest A has NPP is equals to 1254, forest B NPP is equals to 2157 and forest C NPP is equals to 779. Which one of these forests have maximum energy loss by respiration? Give reason. Answer. If GPP is equal, then we can manipulate the NPP equation and solve. NPP is equals to GPP minus respiration of plants. So respiration of plants is equals to GPP minus NPP. This means that the smallest NPP corresponds to the largest respiration, that is forest C. Second, Draw an ecological pyramid of number of the following food chains. A. Grass animal flees on the host animal. B. Trees, insects, woodpecker. Answer A. Grass producer, animal primary consumer, flea secondary consumer. B. Tree producer, insects primary consumer, woodpecker secondary consumer. Section C, question 22. The image below shows two germinated seeds X and Y which belongs to the same species. Seed X is produced by apomixis whereas seed Y is a product of sexual reproduction. Seed X, seed Y. A. Write the number of embryo sacs and ovules in the ovary of seed X. Answer. Seed X 3 embryos, 1 embryo sac, 1 ovule. B. How multiple embryos are formed in citrus fruits? Answer. The nucellar cells grow mitotically and develop into the embryos by asexual reproduction. C. What advantage will plants develop from seed Y have over seed X? Answer. The plants growing from seed X will have to share the resources endosperm, so there is a possibility of some plant being undernourished. Only one plant in seed Y will use the entire endosperm for its growth. Or, as the plants of seed X are clones, they will not show variation and may succumb to environmental stress. Plants from seed Y will have genetic variation and so can show greater adaptability. For visually impaired students, each mango fruit contains one seed. Two mango seeds X and Y were sown in the soil. From seed X, 3 plant saplings germinated, but from seed Y, only one plant sapling germinated. A. For seed X, which is apomictic, calculate the number of first embryo, second embryo sac, third ovules in the seed's ovary. Answer. Seed X 3 embryos, 1 embryo sac, 1 ovule. B. How multiple embryos are formed in citrus fruits? Friends, we have already read the answer of this question. C. What advantage will plants develop from seed Y have over seed X? So we have already read answer of this question too. Let's move to the next question. Question 23. Name the place in human ovary where the first meiotic division is completed during oogenesis. What are the products of this division? Give the chromosome number of each type of cells involved in the process. Answer. The first meiotic division is completed in the primary oocyte during oogenesis. Then primary oocyte undergoes first meiotic division to form a large haploid secondary oocyte and a tiny first polar body. The primary oocyte comprises of 46 chromosomes, whereas secondary oocyte and first polar body have 23 chromosomes each. Question 24. The schematic representation given below shows the concept of central dogma. Central dogma, DNA, transcription, mRNA, translation, protein. And this is replication. A. During the process of replication and transcription, the pairing of nitrogenous bases is not similar. Explain. Answer. During replication, adenine pairs with thymine in DNA. During transcription, adenine pairs with uracil in RNA. B. How is the above process modified in a retrovirus? Name the process. Answer. In retrovirus, the nucleic acid is RNA and it is used to synthesize DNA. The process is called reverse transcription. C. Justify why during the process of transcription only a segment of DNA is copied into RNA. Answer. It is a highly energy rich process, or as per the need only the gene coding for a specific protein is transcribed. For visually impaired students, central dogma explains the process of DNA transcription and translation. From DNA, mRNA is transcribed and then mRNA is translated into a polypeptide. So friends, these questions are similar to the one we have read earlier. So let's move on to question 25. Question 25. Describe the steps involved in southern blot hybridization using radio labeled VNTR as a probe. Answer. Isolation of DNA, digestion of DNA by restriction of endonucleases, separation of DNA fragments by electrophoresis, transferring, that is blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, hybridization using labeled VNTR probe, and Detection of hybridized DNA fragments by autoradiography. Question 26. Biofertilizers are organisms that enrich the nutrient quality in the soil. Explain the role of three main sources of biofertilizers. Answer. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria. The nodules on the roots of leguminous plants are formed by the symbiotic association of rhizobium. These bacteria fix atmospheric nitrogen into organic forms, which is used by the plant as a nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Examples Azospirillum and Azotobacter, thus enriching the nitrogen content of the soil. Fungi are also known to form symbiotic associations with plants. Mycorrhiza. Many members of the genus Glomus form mycorrhiza. The fungal symbiont in these associations absorbs phosphorus from soil and passes it to the plant. Plants having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Example Anabena, Nostoc, Oscillatoria, etc. In paddy fields, cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter to the soil and increase its fertility. Question 27. Explain how PCR technique can be used for amplification of a small amount of DNA template. Answer. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest are synthesized in vitro using two sets of primers, that is, small chemically synthesized oligonucleotides that are complementary to the regions of DNA and the enzymes DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated amplification is achieved by the use of a thermostable DNA polymerase that is isolated from a bacterium, Thermus aquaticus, which remains active during the high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Each cycle has three steps. First, denaturation, second, annealing, and third, extensions. So friends, this is the diagram. Region to be amplified, denaturation, annealing, DNA polymerase plus deoxynucleotides, extension, tag polymerase. 30 cycles amplified 1 billion times. So friends, you can pause the video and note them down. Question 28a, diagram given below depicts different species of warbler birds feeding on different regions on a spruce tree. Explain the mechanism which helps them to coexist. So friends, this is the diagram. Feeding height in meters. Bay-breasted warbler feeds in the middle part of the tree. Cape May warbler feeds at the tips of branches near the top of the tree. Yellow-rumped warbler feeds in the lower part of the tree and at the bases of the middle branches. Answer. They are able to coexist by mechanism of resource partitioning. If two species compete for the same resource, they could avoid competition by choosing different foraging patterns. MacArthur showed that 5 closely related species of warblers living on the same tree were able to avoid competition and coexist due to behavioral differences in their foraging activities. B. What does Gauss's exclusion principle state? Does it apply in the case shown above? Explain. Answer. Gauss's competition exclusion principle states that two closely related species competing for the same resources cannot coexist indefinitely, and the competitively inferior one will be eliminated eventually. No, it doesn't apply to the case above. For visually impaired student, A. Name and explain the mechanism where two species competing for the same resource coexist. B. What does Gauss's exclusion principle state? Does it apply in the above situation? Explain. So friends, we have read answers of these questions just now. Let's move on to the section D. Section D, question 29. Assuming that within a population of beetles where Hardy-Weinberg conditions are met, the color black B is dominant over the color red small b. 40% of all beetles are red small b small b. Given this information, answer the questions below. A. What is the frequency of red beetles? Answer. Given is small b is equals to q square is equals to 0.4. To determine q, which is the frequency of the recessive allele in the population, simply take the square root of q square, which works out to be 0.632. That is 0.632 multiplied by 0.632 is equals to 0.4. So q is equals to 0.63. B. Calculate the percentage of beetles in the population that are heterozygous. Answer. As we know that P plus Q is equals to 1, then P must be 1 minus 0.63 is equals to 0.37. The percentage of beetles in the population that are heterozygous would be 2PQ. So 2 multiplied by 0.37 multiplied by 0.63 is equals to 0.47. Attempt either subpart C or D. C. What is the frequency of homozygous dominant individuals? Answer. Given is capital B is equals to P square is equals to 0.37 square is equals to 0.14. D. Assuming that Hardy-Weinberg conditions are met in the beetle population consisting of 1500 beetles. How many beetles would you expect to be black and red in color respectively? Answer. As 1500 is the total population, 40% is red population. Hence, the number of beetles with red color will be 1500 multiplied by 0.4, and 0.4 is 40 divided by 100. Then we solve it, we get 600. If total population is 1500 and red is 600, then black would be 1500 minus 600 is equals to 900. Question 30. Given below is the pattern of temperature in a person suffering from a non-viral disease transmitted by mosquitoes. Study the graph and answer the questions that follow. So friends, this is the graph. This is temperature in degrees Celsius and this is time days. A. Explain the factors responsible for this pattern of temperature. Answer. The rupture of RBCs associated with the release of toxic substance Hemazoin is responsible for the chills and fever recurring every 3 to 4 days. B. How does this pathogen multiply in the human body? Answer. The parasite reproduces asexually in liver cells, bursting the cell and releasing into the blood. Parasite further reproduces asexually in red blood cells. Released parasite infects new red blood cells. Sexual stages, gametocytes, develop in red blood cells. Attempt either subpart C or D. C. How is this infection transmitted to humans? Answer. The infection is caused by the bite of the female Anopheles mosquito, which introduces the sporozoites in the human body. D. Which stages of the life cycle of this pathogen are completed in the mosquito's gut? Answer. Fertilization and development takes place in the mosquito's gut. For visually impaired students, a non-viral disease that is transmitted by mosquitoes causing recurring fever in an infected person. Explain giving reasons. B. How does this pathogen multiply in the human body? Attempt either subpart C or D. C. How is this infection transmitted to humans? D. Which stages of the life cycle of this pathogen are completed in the mosquito's gut? So friends, we have already read answer of these questions just now. Let's move on to the next section, E. Section E, question 31. Attempt either option A or B. A. Cryptorchidism is a condition in which the testes fail to descend into the scrotum. It can also lead to compromised Sertoli cell function and has an impact on Leydig cell function. First, identify at least 3 parameters of male fertility which gets affected due to cryptorchidism. Answer. Sperm count decreases, spermatogenesis is impaired. Spermatids do not get nourishment to develop into spermatozoa, thus spermiogenesis will be affected. Leydig cells synthesize and secrete androgen hormones like testosterone, so secretion of androgens will be affected. Second, which process will be affected if mature spermatids are not released from Sertoli cells? Answer: spermiation. Third, name and explain one assisted reproductive technology ART process in which the sperm or semen is used to assist fertilization. Answer: artificial insemination AI technique. In this technique, the semen collected either from the husband or a healthy donor is artificially introduced either into the vagina or into the uterus IUI, intrauterine insemination of the female, or intracytoplasmic sperm injection ICSI is another specialized procedure to form an embryo in the laboratory in which a sperm is directly injected into the ovum. Fourth, name and explain the assisted reproductive technology that should be used to complete the development of embryo 1 and 2 shown in the figure given below. So friends, this is the figure embryo 1 and embryo 2, and these are blastomeres. Answer: the zygote or early embryos with up to 8 blastomeres could be transferred into the fallopian tube ZIFT, zygote intrafallopian transfer, embryos with more than 8 blastomeres into the uterus IUT, intrauterine transfer to complete its further development. For visually impaired students, fourth, an infertile couple decided to use ART to conceive. After IVF, they decided for embryo transfer of the following two embryos. Embryo 1 consisted of 8 blastomeres and embryo 2 consisted of 16 blastomeres. Name and explain the techniques they should deploy to complete further development of the given embryos. So friends, we have already read the answer of the same question. Let's proceed further. Or B. First, explain the significance of each of the following features present in plants given below. A. In rose bay plant, the stamens ripen before the stigma. Answer: in rose bay plant, the time of maturation of stamen and pistil is not same. The pollen will not be able to germinate on the stigma. This prevents autogamy in rose bay. B. In certain species of prime rose, the flowers have short stamen and long style. Answer: different position and incompatible placement of the reproductive structure prevent successful pollination and thus autogamy in prime rose. C. The bisexual flower of mustard exhibits rejection of self-pollen grain. Answer: pollen pistil interaction for same species is not possible. This is a genetic mechanism which prevents the pollen grain from forming pollen tube on the pistil of the same flower. Second, explain how autogamy is prevented in castor and papaya plant respectively. Answer: the male and female flowers are present in the same plant but are not in proximity, preventing self-fertilization in castor. In papaya, the male flower and female flowers are in different plants. It prevents autogamy. Question 32. Attempt either option A or B. A. Explain how advent of biotechnology has helped in preventing infestation by nematodes and thereby increasing crop yield. Answer: several nematodes parasitize a wide variety of plants and animals including human beings. A nematode, malloid gene, incognitia, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary dsRNA molecule that binds to and prevents translation of the mRNA, that is silencing. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements, that is transposons, that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded dsRNA that initiated RNAi and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Or B. In the future, genetic therapies may be used to prevent, treat, or cure certain inherited disorders in humans. Justify this statement with a suitable example. Answer. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase ADA deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown into a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. Question 33. Attempt either option A or B. A. First, why are there a need to conserve biodiversity? Any two reasons. Answer. There are three main reasons for conserving the biodiversity which have been classified into the following categories. You can write any two reasons. First, narrowly utilitarian arguments. Human beings derive direct economic benefits from nature like food, firewood, fiber, construction material, industrial products like resins, gums, dyes, tannins, etc., and medicinally important products. Next, broadly utilitarian arguments. Biodiversity plays a major role in maintaining and sustaining supply of goods and services from various species as well as ecological systems. The different ecological services provided are: Amazon forest is estimated to contribute 20% of the total oxygen in the atmosphere on Earth. Pollinators like bee, bumblebees, birds and bats pollinate plants to form fruits and seeds. Aesthetic pleasures like bird watching, spring flowers in full bloom, walking through the thick forest, waking up to bulbul's song, etc., are some other benefits of the ecosystem. Ethical reasons. There are thousands of plants, animals, and microbes on this earth which are not useless. Everyone has some intrinsic value, even if it is not of any economic value to us. It is, therefore, our moral duty to ensure well-being of all the living creatures for the utilization of future generations. Second, name and explain any two causes that are responsible for the loss of biodiversity. Answer. There are four major causes of biodiversity loss. These are also known as the evil quartet. So friends, you can write any two ways listed below. Habitat loss and fragmentation. Destruction of habitat is the primary cause of extinction of species. When large-sized habitats are broken or fragmented due to human settlements, building of roads, digging of canals, etc., the population of animals requiring large territories and some animals with migratory habitats declines. Overexploitation. When biological system is overexploited by man for the natural resources, it results in degradation and extinction of the resources. For example, stellar sea cow, passenger pigeon, and many marine fishes. Alien exotic species invasions. Some alien exotic species, when introduced unintentionally or deliberately, become invasive and cause harmful impact, resulting in extinction of the indigenous species. Nile perch, a large predator fish, when introduced in Lake Victoria, East Africa, caused the extinction of an ecologically unique species of sechiled fish in the lake. Co-extinctions: when a species becomes extinct, the plant and animal species associated with it in an obligatory manner also become extinct. For example, if the host fish species become extinct, all those parasites exclusively dependent on it will also become extinct. Or B. First, name the two types of desirable approaches to conserve biodiversity. Explain with examples bringing out the difference between the two types. Answer: in situ conservation. It is the conservation and protection of biodiversity in its natural habitat. Ex situ conservation. It is conservation of selected threatened plant and animal species in places outside their natural habitat. In situ conservation: population is conserved in the surroundings where they have developed their distinctive features. Ex situ conservation: population is conserved under simulated conditions that closely resemble their natural habitats. In situ conservation: example, national parks, biosphere reserves, wildlife sanctuaries, etc. Ex situ conservation: example, botanical gardens, zoological parks, wildlife safari, gene banks, etc. Second, state the features of a stable biological community. Answer: a stable community should not show too much variation in productivity from year to year. It must be either resistant or resilient to occasional disturbances, natural or man-made, and it must also be resistant to invasions by alien species. Friends, our sample question paper for class 12 biology is complete. If you like the video, kindly like it and share it with your friends. Thank you for watching.